Okay, so let's get started. So welcome everyone to logging to the museum during the weekend. It's really my pleasure to welcome today's speaker, Dr. Lifan Lu from UCSD, also my friend. So Lifan currently is a full professor in Department of Molecular Biology, School of Biological Sciences in UCSD. Just a very brief introduction of Lifan's background. So Lifan completed his undergrad study in zoology in Taiwan, in National Taiwan University, and then continued to pursue his PhD in immunology in Dartmouth Medical School. And then after which, from 2006, he started to his postdoc training in immunology in Memorial Sloan Kettering with also in University of Washington. I think first in University of Washington and then Memorial with Sasha Rudansky. So from 2011, Lifan started his independent research program as assistant professor in UCSD all the way to today as a full professor in School of Biological Sciences, UCSD, as I just mentioned. Lifan's lab major research focus are to try to identify the molecular machinery of differentiation and function of Treg cells, and then to ultimately gain insight into the complex biology of Treg cells, which including the maintenance of immunological tolerance to self, and then the regulation of immune response to pathogen, commensals, and tumors. There are two major focus in his lab. One is try to identify microRNA, which is crucial for mediating Treg cell biology in both healthy and disease status. And also the second one is try to elucidate molecular and cellular machinery by which Treg cells control type 1 inflammation. So with really productive PhD and postdoc training, also during his own lab independent research, he actually really come up with very productive period, and then received numerous honors and awards, which including 2007 Leukemia and the Lymphoma Society Fellow Awards, K989 from 2011. 2012 get Sidney Kimmel Scholar Awards, 2013 Helmer Fellow Awards, and then 2016 as a Pfizer Showell Travel Awards. So I think today Lifan is going to talk about some of his lab's recent work, which focused on the cytokine IL-27. And then with that, thank you, Lifan, very much for contributing the lecture for us. We are very much looking forward to your talk. Please go ahead.
Thank you. So everybody can hear me, right? So yes, I want to thank Tran to give me this opportunity to talk about some of the work that we have been working on in the past few years about the role of IL-27, particularly in regulating intestinal homeostasis. And I'm sure that for the audience today, I don't need to go through too much details about the background introduction, but I just want to quickly mention that the major research interest in my lab is to understand immune regulation. As we know, if you have too much or too little, would all lead to unwanted pathological outcome. So in particular, in my lab, we are interested in study T-cell immunity. As we know, T-cell develop in the thymus, and those naive T-cell, when they're in the periphery, encounter antigen-presenting cell, dendritic cell presenting antigen, plus the signal that dendritic cell provide, depending on the type of pathogens that those dendritic cells encounter. those naive T cells are able to differentiate into different helper T cell lineages through acquiring different transcription factors, allowing them to produce different effector cytokines to fight against different type of pathogens. And in the gut, in particular, we know is a very specific environment where you have both commensal as well as potentially dangerous pathogens like Toxoplasma gondii, Citrobacter rodentia, among others, could invade our body because the gut lumen where they reside is really just one layer of cells away from our body. So inside, underneath this intestinal epithelial cells layer, we have different T cells, obviously, to protect us from those potentially invading pathogens. And as you can see in this cartoon figure, there are Th1 cells, Th17 cells that can produce interferon gamma and IL-17. And at the same time, we also know that you cannot have too much of immune response because that can cause immunopathology. So in this case, early on, there was identification of a subset of T cells that are good at producing cytokine IL-10, and those T cells were called type 1 regulatory T cells at the time, TR1 cells, which is critical to control those effector T cell responses. Subsequently, the identification by our lab as well as others to show their specialized phospholipid positive regulatory T cells, like Tbet positive Treg cells, that are able to control Th1 cells specifically. And there's other studies demonstrating, of course, like STAT3 positive phospholipid positive Treg cells are able to control IL-17 producing Th17 cells specifically. So those are the cellular players, but they are also molecular players. And in particular, in today's talk, as I mentioned in the first slide, IL-27 is the molecular player we're going to talk more today. So IL-27 has been studied for a long time. Initially, it was identified as a Th1-like cytokine like interferon gamma because it also lead to the step 1 phosphorylation and the Tbet upregulation. But what later was shown was instead of immune stimulatory, IL-27 is more immune regulatory. Because in the IL-27 deficient mice or IL-27 receptor deficient mice, I'm talking about the germline deficient mice, what people found is they are incapable of producing IL-10. Also, we have shown, as well as other lab, like Chris Hunter's lab at UPenn, show that IL-27 signaling is also essential for the Tbet positive Treg cell differentiation. So in the absence of IL-27, what you are going to get is this dysregulation of interferon gamma and IL-17 responses. So again, as I said, IL-27 was initially identified as a Th1-like cytokines, but it actually belonged to this IL-6, IL-12 cytokine family. And there are many members in this family. They all kind of compose of different subunit together to form a cytokine. For example, IL-27 consists of two subunits, EBI3 and P28. And it was thought to be mostly produced by myeloid cells like dendritic cells and macrophages. And as you can see in this cartoon figure from the review article back in 2015, I already told you IL-27 was shown to be important to induce Tbet positive Treg cells. It's important to induce IL-10 producing TR1 cells. It can also directly induce expression of inhibitory receptor like PD-L1, PD-1, CD8-4, and also inhibit helper T cell function. So it's immune regulatory cytokines that have multiple functions. So when we talk about cytokines, you always think about this is a soluble factor, right? So when you talk about soluble factor, you wonder, does it matter which cell produces them if it's soluble, right? In fact, if you go back to the textbook, when we talk about cytokine, there are three major ways they can function. There's autocrine action. So it means that the cell that produce cytokines act on the cell that producing it. And then you have the paracrine action. It means the cell that produce it, those factor, soluble factor, will act on the neighboring cells. And then you have the endocrine action. It means that the soluble factor like IL-27, for example, if it's work at the endocrine action, it means that it can actually travel and act on distant cells. So if it's act as an endocrine action, then you will probably think it doesn't matter which cell produce it. But what we have shown actually back in 2015, that we already show that actually it matters which cell produce it. For example, in terms of Tbet positive Treg cell differentiation, we know in the absence of IL-27 at all, right? In the germline null mice, you don't get Tbet positive Tregs. But when you try to delete IL-27 in different cell subset, what we found is only when you delete IL-27 in the dendritic cells, then you lose Tbet positive Treg cell differentiation. This is at steady state, so you don't get a lot of Tbet positive Treg cell at steady state and wild type, but you can see about 5-6%, and it comes down quite significantly if you don't have IL-27 produced by dendritic cells. As a consequence, and during toxoplasma infection, where type 1 immunity is important to clear the pathogen, but at the same time, the interferon gamma responses need to be controlled by Tbet positive regulatory T cell, or you are going to get interferon gamma driven immunopathology. So what we found is if you don't have IL-27 produced by dendritic cells, you get less Tbet positive Treg cell, you get dysregulated interferon gamma responses, and then you get enhanced immunopathology. So this is good, but as I told you, the effect is specific because when you delete IL-27 in the germline manner, you get multiple phenotype. But when you delete IL-27 only in dendritic cell, you get reduced Tbet positive rate, but you don't get loss of IL-10 production. So the T cells can still produce IL-10 normally, the TR1 cells. So just to show at the moment is we have the Th1, Th17 cell, and we have different regulatory cell subset doing their job, but it's saying the dendritic cell derived IL-27 is specific for Tbet positive Treg cell differentiation. So how about IL-10 production then? So we know Th1 cell, one of the major functions for Th1 cell is to help macrophages. So it is possible that macrophages can now come back to induce TR1 cells as a regulatory feedback mechanisms. So we wonder whether macrophage-derived IL-27 is the key to drive the IL-10 production in TR1 cells. So that's what we're going to test. And indeed, when we crossed IL-27 P28 flux mice to lysozyme-free mice, we are able to see the reduction of IL-10 production. This could not be found in the DC specific deletion. But we don't see any difference in Tbet positive Treg differentiation. So basically, that to put this cartoon figure together is we have the dendritic cell derived IL-27 specialized to induce Tbet positive Tregs. And then we have macrophage derived IL-27, which is required to induce IL-10 production. Now, let's take one step further. In the gut, as I told you in the early slide, the gut lumen and the lamina propria is separated by a layer of cells, the intestinal epithelial cells. And for long times, it has been shown there's another specialized CD4 T cell subset in this compartment. And what makes them special is they actually start to express CD8. So this CD8 alpha positive CD4 positive IEL has been shown to play also an immune regulatory role. So if you don't have this population, as shown in this Nature Immunology paper back in 2012, they actually have more severe inflammation and colitis. Okay, so there's another immune regulatory cell subset reside in the intestinal epithelium. Okay, so and those are CD4 T cells that differentiate into CD8 alpha CD4 IEL when they move into this specific microenvironment. Now, it was shown that this population required Tbet expression for their differentiation. And in vitro, as shown in this immunity paper, both interferon gamma and IL-27 can induce this Tibet-driven CD8 alpha CD4 IEL differentiation. However, in vivo, in this paper, what they show is only interferon gamma can do that, because when they delete IL-27, they don't see any defect. Only when they delete interferon gamma, they see a reduction of CD8 alpha CD4 IEL. But the key things that they didn't do in that immunity paper back in 2014 is they look at early days, like day 80, okay, like 10 weeks after birth. As I told you, that this population differentiate into the intestinal epithelium over time and accumulate over time. So actually, at 10 weeks after birth, they haven't reached their plateau. So if you track them over time, actually only after day 90, day 100, then you reach the plateau. So we revisit this question. So we cross the IL-27 receptor flux mice. So now it's not a cytokine, it's a receptor to CD4 T cell to create a T cell specific receptor knockout. And what we found, yes, in the young mice, as suggested in the immunity paper, we don't see any difference when we delete IL-27 receptor in T cells. However, in the old mice, you can see in the wild type mice, the CD8 alpha CD4 T cell keep accumulating, reach their plateau about 60%. While in the T cell receptor knockout, they stay at 40. So this suggests that IL-27 signal in T cell is still required for CD8 alpha CD4 IEL differentiation. So we look at the mice that couldn't produce IL-27 in specific subset and find out then dendritic-derived IL-27 is dispensable. As you can see here, they get 60%, just like the wild type. Myeloid cell-derived IL-27 is also dispensable. As you can see, the same frequency of CD8 alpha CD4 IEL. So what other cells can produce IL-27 that is needed? So in the past, there was a study back in 2011 showing actually in the lung epithelial cells can actually produce IL-27 upon TLR receptor stimulation. So we wonder whether the gut epithelial cells can also do the same thing. So to do so, we cross the P28 flux mice to the Villing-1 Cree. So we remove IL-27 production by the gut epithelial cells. And this is just to show you, indeed, we can see gut epithelial cells to produce IL-27 by qPCR. So both IL-27 P28 go up, EBI3 go up, but when you delete the P28 by crossing to Villing-1 Cree, you can see the P28 is abolished, while the EBI3 remain the same. So this is a qPCR. So then we look at by ELISA, look at IL-27 protein production. Again, in the Villing-1 Cree, the IEC specific knockout, you could no longer detect IL-27. Well, you can still detect IL-27 produced by the IEC in the CD11. Well, sorry. So when you can still detect IL-27 produced by the dendritic cells as well as the macrophages in the IEC knockout mice. So this is specific knockout. But as you can see here, when we remove IL-27 production by IECs, now you don't see the accumulation of CD8 alpha CD4 IEL in those mice, similar to what we observed in the T cell receptor knockout mice. But this IEC-derived IL-27 is dispensable for Tibet positive Treg cell differentiation. It's also dispensable for IL-10 production. So as you can see, they still have the same IL-10 production, same Tibet positive Treg cell in the IEC knockout. So now you wonder, what are the biological consequences if you have this reduced CD8 alpha CD4 IEL in IEC knockout? So it was shown that actually the CD8 T cells, the TRM or resident memory cells, are required to protect the mice from toxoplasma infection. So this is in this cartoon figure, the CD8 positive IEL, the TRMs is important. So how about this Tibet positive CD8 alpha CD4 IELs? I mean, it has been shown they have an immune regulatory role, but can they also play a role in the pathogen protection? or it just simply play a role in controlling immune responses, right? So we wonder what happens if you don't have enough. So we first look at interferon gamma production during toxoplasma, the infection. We don't really see, so this is at the steady state. Okay, so at the steady state, we really don't see much of interferon gamma production change. So it feels like it's not like what we found in the DC knockout. However, when you infect them with toxoplasma, then you now can see uncontrolled toxoplasma burden, both in the small intestine as well as in the liver. And if you use a specific toxoplasma strain where it has the TD tomato, you can track by flow. You can also see in the gut epithelial cells in the IEC knockout, there's this increased toxoplasma burden parasite in the gut epithelial cells. So it seems that this population is actually required to provide protection against the toxoplasma gondii infection. But this is still an indirect evidence because what we did was to remove IL-27 production by IEC. So it could be not due to the effect through the CD8aa CD4 IEL, but through the other effect that is IL-27 dependent. So to really prove this phenotype we observe is due to the reduction of CD8aa CD4 IEL, we performed the reconstitution study. So basically, in the wild type and the knockout mice, we transfer CD8aa CD4 IEL. And as you can see here, in the wild type, you get about 60%. In the wild type with the transfer, it doesn't go further up, it's already reached the plateau. In the IC knockout, you get about 40%. But if you transfer CD8aa CD4 IEL, you are able to make the number go to the plateau level. And this is a statistic. So we are able to rescue the number by supplying more CD8 alpha CD4 IEL in those mice. And as a consequence, now the pathogen burden is now under control. So in the IC knockout, if you are able to supplement with more CD8 alpha CD4 IEL, now you are able to control the parasite better. So now, in addition to the dendritic-derived IL-27, macrophage-derived IL-27, now we introduce another player that intestinal epithelial cells produce IL-27, which add on CD4 T cell to promote the CD8 alpha CD4 IEL differentiation, particularly at the later stage, which is also critical to provide protection against pathogens, gut pathogens. Okay, so now I'm going to move on to the study that we haven't really published. So those work I told you has been published. So now I want to move on to a more specific story about Tregs. So I just told you that different T helper cells can differentiate to control different type of pathogens. But at the same time, if they are dysregulated, you also have this different T helper cell driven autoimmunity. And since 2009, several studies have shown that Treg cells have also the ability to acquire differentiation factors, allowing them to control different type of T cell responses. Also, depends on where they are in different tissue microenvironment, they can also have a special functional role, allowing them to better control this environmental specific immune response. Like the study shown in the muscle, in the adipose tissue, of course, in the gut, you have this specialized Treg cells. They are optimized to do their job in this local environment. Now, the question has remained, still remain is, there's a lot of sequencing study to compare different Treg cells from different tissues. However, in those studies, they are not really study Treg cell coming from the same animal when they are having this type of different disease condition. For example, the muscle Treg cell was studied using a muscle injury model. The fat Treg cell was using the mice with obesity. So you can always argue that the direct comparison between Treg cell from the inflammatory environment is still missing. So to address this question, we took advantage of this dt563 DTR model, which we are able to induce the multi-organ autoimmunities by giving diphtheria toxin, leading to a temporal Treg depletion. And that you can, if you keep giving DT, you're able to remove all Treg cells and the mice will die within 3 weeks after you give DT. However, if you transfer congenitally marked Treg cells in time with sufficient number, you are able to rescue this multi-tissue organ autoimmunity. And this is the protocol that we employ. And we know that if you transfer too early, then you don't get enough inflammation. If you transfer too late, then the mice are already too sick to be able to be rescued. So you have to transfer around, certain time, and then you also don't want to wait too long, or the mice will be completely healthy. So we take out the mice around day 10, 10 days after Treg transfer. And this is just to show you how it works. In the control mice, obviously you have Treg cells showing the FOXP3 positive populations. They express KIC7 as a marker for proliferation. And then you have CD25 also can serve as an activation marker for conventional T cells. So in normal condition, the conventional T cell proliferate and have not much of activation. But if you remove Treg completely by DT treatment, the conventional T cell proliferate like crazy and they are hyperactivated. But if you put the Treg cell back in, then you can see the proliferation of conventional T cell drop and the activation of conventional T cell also drop, suggesting that the Treg cells are doing their job to keep the mice healthy. So we are able to isolate the Treg cell from those DT-treated animal and using the congenitally marker, and then to set up RNA sequencing analysis from different tissues. So in this case, we can compare both healthy and Treg cell isolated from a healthy environment versus a sick environment in multiple tissues. And I just want to give you one example. So we focus on the small intestine, for example, in this study, is if you look at this scatter plot, you can see the x-axis is Treg over Treg in the healthy control animal with PBS injection. And in the y-axis is the Treg over T effector cell, conventional cell in the DT-treated animal, so in the inflammatory environment. So what we are interested is looking at this red box, the genes that only go up or only go down in Treg cells in the sick environment, because we already know what Tregs are, what kind of difference they are compared to conventional T cell in a healthy condition. But what we are interested in knowing is whether in the inflammatory condition, when they actually have to work much harder to rescue those mice, whether there's specific genes that can be identified. And then you do the Venn diagram analysis to compare those genes from different tissue microenvironment, and you can look for tissue-specific gene signature. I mean, of course, you can do a lot of bioinformatic analysis, so I want to quickly jump to today's target, IL-27. So what we found is interesting, is it has been known for a long time, EBI3 is highly expressed in Tregs. So you can see in the Treg, in the control mice, EBI3 is already high. Now, P28 was never really detected in Treg in the previous sequencing studies. However, you can see here is in the gut, in the Treg, isolated from the sick environment, now P28 can be induced. Okay, so as I told you, P28 combined with EBI3 give you IL-27. But what is interesting is in the past, because it was only EBI3B detected, while B28 was never detected in Tregs, so it was suggested EBI3 pair up with B35 to form IL-35. That is the cytokine that Treg produce, because they all share EBI3. So in this study done by Dario Vignali back in 2016, so they show by creating an EBI3 flux mice crossing to FAST3, they show that Treg cell derived IL-35 is important for their function, in particular in the tumor setting. So that makes us wonder, because when you don't think P28 can be expressed in Treg, P35 combined with EBI3 from IL-35 is the molecule. But now, since we know IL-27 P28 can be induced in gut Treg under inflammatory condition, that we wonder, perhaps in those EBI3 flux mice, some of the phenotypes are not completely attributed to IL-35. Maybe IL-27 is also part of the player in the Treg mediated immune regulation. In fact, we confirm our study by qPCR. Again, you can see P28 is induced in gut Treg, isolated from the DD treated mice, but we couldn't detect beta-35 in the gut Treg from the same DD treated mice. So it feels like the IL-27 produced by Treg is probably more important, at least in our gut disease model. So to study that, we again cross the IL-27 P28 flux to the FAST3 mice to create a Treg specific knockout. And in general, the mice are healthy. So the mice, you don't really see very overt immunopathology. If you look at gut, the activation, the proliferation of effector cells are consistent, comparable between the wild type and the knockout. But the only phenotype that we saw is increased IL-17 production by the T converter T cells in the gut. Okay, so it's not in the spleen, it's only in the gut. And we don't see other cytokine, like interferon gamma or other cytokine being dysregulated. It's only IL-17, and I show IL-17A here, but there's also IL-17F and also GM-CSF. So it's IL-17 related family cytokines go up. But the mice are generally healthy, so you see this increase, but the mice are not really autoimmune. So we wonder what happened if we remove IL-27 from other cells. So because I just told you Treg derived IL-27 is important, but I always show you in the early slide that IL-27 can be produced by other cells. So can they also contribute to control Th17 response? Because we didn't check that earlier. So we look at that. We look at back to our myeloid specific knockout, IEC knockout, dendritic cell knockout. Again, we don't see any change in Th17 response. So if you really put all this data together, you can see the myeloid knockout, DC knockout, IEC knockout, the IL-17 response compared to the wild type is the same. The Treg knockout, you have increased IL-17 response. Same thing as the T cell receptor knockout. So this suggests that IL-27 produced by Treg act on T cells is important to inhibit IL-17 responses. Whether or not it's STAT3 positive cell, we don't know, but we know Treg produce IL-27, which is specific needed to control Th17 response. But what are the biological consequences? Because I told you the mice are generally okay. So we first employed this anti-CD3 induced colitis model, which was shown by Richard Favell many years ago by injecting anti-CD3 IP in the mice, 3 shots, that you induce inflammation in the gut specifically that is Th17 driven. So as you can see here, in the wild type mouse, you have the weight drop, but in the Treg knockout, you drop weight even further. And you get more severe pathology. And also, if you look at the IL-17 response, it's much, much more higher, right? In the wild type, you get about 14%. In the knockout, now you get about 30%. But if you look at spleen, there's no clear difference. Now, that's acute. So how about in the chronic situation? So we employ another model, which is ALN, a carcinogen, DSS-induced colon cancer model. So basically, if you give 1 shot of carcinogen plus chronic inflammation, that will lead to the development of colon cancer, which have also been shown to be IL-17 driven. So as you can see here, by giving this protocol, 1 shot ALN and then 3 cycles of DSS treatment, wait for 12 weeks, and you harvest the mice, and you can see the wild type mice, you get colon cancer formation, but in the Treg knockout, you get much more. And again, you see increased IL-17 response and increased pathology, both in the tumor infiltrating cells as well as in the neighboring intestinal tissue. So now in both chronic, acute and chronic model, we have shown this Treg-derived IL-27 is important to control Th17 responses. Now, you could argue that, of course, you didn't see the difference in the spleen because all the disease model you have shown is the intestinal disease model. So if you induce a Th17 response outside the intestine, perhaps the Treg-derived IL-27 will also be important. So that's not an intestinal specific feature. So to do so, we employ the EAE model, which we also know Th17 play a role in addition to Th1. And it also has been shown IL-27 play a role to inhibit Th17 in the EAE model in the central nerve system. So we test that, but first confirm the previous study using the IL-27 germline knockout. Now we use a receptor knockout, but specifically in T cells. So when we make T cells incapable of responding to IL-27, indeed, the mice are sicker, so you get more enhanced disease score, and you also get higher IL-17 response. I just showed you IL-17A here, but the same thing for IL-17F and GSSF. So you need IL-27 to inhibit Th17 response in the EAE model, consistent with what people showed before. But if you look at the Treg knockout, no difference. So in the Treg knockout, the disease score the same, the Treg knockout, IL-17 response remain the same. So this is consistent with our model is IL-27 is produced by gut Treg, is specifically needed for Treg to control gut Th17 response, not in the other tissue microenvironment. Okay, that's all bad when you have over or uncontrolled Th17 response, but we also know Th17 immunity is needed to provide protection against pathogen, like seizure vector retention. This is a bacterial pathogen that requires Th17 immunity to protect the host. So what happens if you increase the Th17 response? Again, in the Treg knockout, you infect the mice with seizure vector. Indeed, you get enhanced Th17 response. Consequently, you also see reduced bacterial burden. So enhanced Th17 response can be beneficial in the Treg knockout condition. In this case, is enhanced Th17 help to clear the pathogens. Now, the question is, okay, because you infect the mice with the Th17-driven pathogens, what happens if you infect the mice with another pathogen that drives other type of immune response? Does Treg cell-derived IL-27 play any role? So we go back to our T-Gandia infection model, which we know it drives Th1 response. You need Th1 response to provide protection. So in this case, deletion of IL-27 in Treg has no effect. You still get the same level of gamma, because we know interferon gamma production need Treg, T-bed Treg to control them, but T-bed Treg is developed depending on the IL-27 produced by dendritic cell, as I showed you earlier. Now, IL-27 produced by Treg cell have no impact on regulating the interferon gamma response. And also, there's no impact on the pathogen burden. So the pathogen can be cleared equally. So to put this together, so far, I show you the Treg-derived IL-27 is specifically needed to control Th17 response in the intestine. Now, you will question, do all Tregs cells produce IL-27 in the gut? Or is there a specific subset of Treg cell in the gut produce IL-27? Because we know Treg cells are heterogeneous. So we perform the single-cell RNA-seq to study the intestinal Tregs in the gut in the inflammatory setting. In this case, we use a seizure vector retention infections model. So we can basically largely separate into 4 clusters. And if you look at the genes that are enriched in this full cluster, you will see the cluster 0, R0, have the genes that resemble activated Tregs. And you have the R1 cluster that resemble effector Treg signature. And you have the R2 cluster that resemble resting Treg signature. And the R3 is kind of ST2 positive Treg cell because ST2 is one of the genes that show to be enriched in this cluster, even though it's not that different. Okay, but it is the population have ST2 expression. So how about IL-27? Unfortunately, for single-cell RNA-seq, it's not a good method to detect cytokines, as some of you may have experience working with single cells. So for low abundant genes, normally you just hard to detect them. So single cell approach is good to detect those high expressors, but for low abundant genes like cytokine, usually it's hard to detect them. So we can only have a few dots, okay? So it looked like noise, but I want to show you this is not noise, okay? So it looked like IL-27 P28 is only shown in R0, while EBI3 shown in both R0 and R1. And remember, I told you R0 is an activated Treg signature, which have CD8aa, also express TCF7, but also TCF7 is also expressing R2, which is a resting Treg population. But in the R1 is affected Tregs that have high level IL-10, high level granzyme B, high level ID2, high level of all those affected molecules, FGO2. In R2, the resting Treg, you have high level CD62L, high level of BAC2, as I told you, also TCF7. So those genes resemble those kind of expressing those different clusters. So we want to confirm whether the IL-27 production is restricted to R0. So to do that, we take advantage of the cell surface molecule, CD83 and CD62L. So if you look at the CD83, CD62L expression pattern, you can largely separate the three populations. R1, which is CD83 low, but CD62L low, double low. R0 and some of the R3 are mainly CD83 high, but CD62L low. And then you have the CD83 low, but CD62L high R2 population. So when you separate these three populations and we confirm by qPCR, as you can see, P28, IL-27 P28 expression is restricted to R0 and maybe some of the mixture of R3. And you don't see P28 really in R1 or R2. EBI3 is the one that from single-cell RNA-seq we can detect in both R0 and R1. Again, you can see it's both expressing both R0 and R1. And IL-10, on the other hand, is restricted to the R1 affected Treg population, and we can reconfirm this data by qPCR using the FACS sorted cells. So to summarize what I have shown you now is we show the unique transcriptome signature in tissue-specific Treg cell under different cellular environmental contexts. In particular, IL-27 is specifically secreted by intestinal Tregs under inflammatory conditions. And Treg cell-specific IL-27 P28 ablation led to elevated Th17 responses selectively in the gut. And loss of Th17 controlled by intestinal Treg cells incapable of producing P28, IL-27, could be both detrimental or beneficial in a context-dependent manner. Lastly, we have shown there's a subset of Treg cell which express CD83, seems to play a major role in IL-27 production. So now, go back to the initial slide I told you. Are all IL-27 created equal? Based on our study, we believe it's not. So it suggests that IL-27 mainly function as in the paracrine action manner that act directly to their closely neighboring cells. So even though it's soluble, but it is important which cell produce it and which cells are nearby. Those really dictate the outcome of IL-27 dependent phenotype. So this is our current model. that we have shown that IL-27 can be produced by dendritic cell, which is important for T-beta positive Treg differentiation to control Th1 response. Which also showed IL-27 produced by macrophages is important for IL-10 producing TL1s. Now, I also show you IL-27 produced by IECs is important to promote differentiation of CD8aa CD4 IEL, which also provide protection against intestinal pathogens. Lastly, I show you CD83 positive Treg cells are likely specialized to produce IL-27 under inflammatory condition in the gut to specifically control IL-17 responses. So I want to thank people in the lab, especially Jia Hao Ling, the picture shown here, who was a postdoc in my lab, worked on this two project, IL-27 project, both the CD8aa CD4 IEL project as well as the Treg project, and other member in the lab. And also I want to thank in Zhinan specifically to provide IL-27 P28 flux mice, allowing us to be able to initiate this IL-27 project since very early on, 2014. And also I want to thank Bu Qiming, who is a professor at Northwestern, who provide the IL-27 receptor flux mice, so we can also study the cells that respond to IL-27. And of course, I want to thank my funding resource, and I will be happy to take questions. Thank you.